Welcome viewers. Today we will be discussing about the practical induction by IPTG leading to overexpression of protein in E. coli. Overexpression in E. coli or overexpression of a protein is supposed to be one of the most remarkable developments in the field of biotechnology. Now, the simplest definition of biotechnology is use of biological processes in industry and technology. In 20th century, it gathered maximum momentum with the finest discovery by Alexander Fleming. In the year 1929, he for the first time discovered penicillin and isolated it from the mold Penicillium, and this drug was used for killing the bacteria. This remarkable discovery opened the doors, and there came many other groups, many scientists. They discovered many other important metabolic compounds from bacteria and other microbes. The journey was moving on smoothly when a hurdle came, and that hurdle was that pharmaceutical companies could find that there are various other important compounds that were obtained or that can be obtained from eukaryotic cells. So how we can overproduce these metabolic compounds or important compounds which can be obtained from eukaryotic cells. Then emerged the story of cloning and expression of a gene, wherefrom we started cloning and expressing a eukaryotic gene inside a prokaryotic host, say bacteria. This journey of overexpression started from that point. A unicellular organism is induced to produce a disproportionately large amount of protein. This is supposed to be the basic definition of overexpression. Now, what are the benefits of overexpressing a protein? If we overexpress a protein, that can be used to study a protein meticulously. Second important thing is it helps us to assist or cure many diseases. As for example, the diabetes can be cured by providing insulin, and bacteria are used for overproduction of insulin. Third, it helps us to determine the various enzymatic mechanisms which are undergoing inside any living system. And finally, it helped us in producing many life-saving products. So let's have a look into the background of gene expression, that what happens during gene expression. The DNA which gets transcribed to form an mRNA, this is supposed to be the transcription step. After that, this mRNA is being read or is being converted into proteins during the process of translation. So these two factors or these two events together form gene expression. Now, if we consider about gene expression, we can see that there can be two types of genes which exist in any living system. Some of them are called constitutive genes, which are synthesized throughout the life cycle of the bacteria, but they are being expressed by very weak promoters, and as a result of which their expression level is very low. On the other hand, there are certain other genes called inducible genes, which are synthesized only under specific conditions, and these are being synthesized by inducible promoters, which are very strong promoters, and hence the number of transcripts that they can generate per unit time is much higher. So if we desire to overexpress any protein, then we will prefer to look into or to focus on these inducible promoters. The essential prerequisites within an expression vector is called as a bacterial promoter, wherein the RNA polymerase will bind and carry on the transcription, as I said, which is the first step of gene expression. Then there exists an operator sequence which is responsible for switching off the gene expression when it is not needed. It means this promoter is an inducible promoter because of the existence of this operator sequence. Next important thing that is present is the ORI region or origin of replication, which is needed for the replication of the plasmid. And the next important part is called as the transcription terminator sequence. If the gene gets transcribed, it has to undergo transcription termination, which is being done utilizing these sequences. Then after transcription, we need the gene to be translated, which is being done taking help of ribosome binding site, which are the sites which are being recognized by the ribosomes to carry on the translation of the gene. And this is the important site called polylinkers region, wherein we are going to introduce gene of our interest, which will be overexpressed inside this expression vector. And finally, we need to have a selectable marker. This is very important as it helps to select only those bacterial cells that are harboring this expression vector, or that are harboring the expression plasmid with the insert. So now let's have a look that there are certain factors that are affecting the level of expression of a clone gene. Number one is promoter strength. We will be describing it soon. Next, we have got a transcription termination signals should be there. Then the plasmid compete number should be high to enhance the expression level, and stability of the plasmid is to be maintained to maintain the expression of the gene. Next, the host cell physiology, that certain important features should be incorporated inside the host so that it can be used to allow the expression of the expression vector. Next is, we should have translation initiation sequences, as I said, and codon choice is a very important point, as the codons which are coding for amino acids, the corresponding tRNAs should be present within the host cell. Next, the structure and stability of the mRNA is very important, as because if too much of secondary structures are formed, then the translation event will be hampered. And finally, last but not the least, the stability of the protein is very important. If we have expressed the protein, that is not mere enough, it should be stable enough. Any inducible promoter that hits our mind first is supposed to be the Lac promoter. It is an inducible promoter wherein we already know that it comprises of three structural genes, namely Z, Y and A, that codes for beta-galactosidase, permease and trans-acetylase respectively. This is an inducible promoter. There also exists a constitutive promoter that continuously synthesizes a protein called repressor. And this repressor protein has got the tendency to bind to the operator region. It binds to the operator region and prevents the binding of RNA polymerase onto the promoter region, as a result of which the structural genes are not synthesized in absence of inducer. So who is the inducer of this operon? Lactose is the inducer of this operon. As soon as lactose comes into the system, it binds to the repressor, inactivates the repressor, it leaves the operator site, and what we see here that the promoter site is vacant. It is being occupied by RNA polymerase and structural genes are getting switched on. So this is supposed to be the best operon which can be utilized for overexpression of any gene inside E. coli host cells. So what recombinant biotechnologists have done, that they have utilized this Lac promoter, operator and repressor system for expressing any gene of interest. They have deleted this Z, Y and A genes, and in this place we can introduce any gene of our interest inside any expression vector. So we have discussed that lactose operon can be used for overexpression of any gene utilizing the Lac promoter operator system and the Lac repressor gene. Instead of lactose, nowadays we use IPTG as the inducer of Lac operon.
And what is the advantage of using IPTG over lactose?
IPTG is supposed to be a gratuitous inducer. It means this is not going to break down by the enzymes which are being synthesized by the Lac operon, unlike lactose. As lactose breaks down by the action of beta-galactosidase, but IPTG doesn't do so. So it remains in the system throughout and we don't need to supply repeatedly IPTG. The major hurdles during overexpression of a protein is that sometimes the proteins that we are trying to express is severely toxic to the host cell. Second important problem is the protein that we are getting may be insoluble, so we need to solubilize the protein. Third hurdle is the secondary structures in the mRNA sometimes restricts or inhibits the rate of translation. Then there are certain proteins which are rich in codons and their corresponding tRNAs are missing in the host cells. Another problem is uncontrolled basal expression sometimes leads to hindrance in the life cycle of the host cell. Next, sometimes too much of induction or overproduction of a protein leads to the protein getting into the inclusion bodies, or they are getting locked within the periplasmic space. So these are the major problems that are being faced during overexpression.
Ma'am, what are the important features that should be present in a host strain used for overexpression?
The strains which are being used for overexpression of a protein should be end A minus. It means it should not synthesize endonucleases. Then the DNA which is getting inside the host cell will be stable enough. Second important point is that it should have a mutation in the REC-A1 gene. It means it abolishes homologous recombination, as homologous recombination may lead to some changes in the gene if it gets integrated within the host chromosome or if it gets altered because of homologous recombinations. Third important thing is that the strain should be lacking of proteases like OMPT protease or LON proteases, as proteases will chop up the protein that we are expressing. And finally, it should be free of F' episomes, as we have seen that in F' episomes many a times contains various protease containing genes. Next is the model vector that is being used for overexpression of genes in E. coli is a PET expression system. So what are the features that are present inside the PET expression system is that we have got a T7 promoter, we have got Lac operator, and this P-link is the multiple cloning site wherein the gene of insert will be introduced. We have got antibiotic selection marker as ampicillin resistance cassette, and Lac-I gene is there to synthesize the Lac repressor protein.
Ma'am, from where do we get T7 RNA polymerase to express the gene of protein using PET vectors?
It's a very important thing to understand, as wild type E. coli never synthesizes T7 RNA polymerase. So they have done genetic engineering in the genome of wild type E. coli. Thereby they have introduced the T7 RNA polymerase gene into the downstream of the Lac promoter. The host that is being used for overexpression of any gene using this PET vectors is named as BLDE3. Herein you can see that in the downstream region of PLAC we have got T7 RNA polymerase gene, and this T7 RNA polymerase won't be synthesized constitutively. It will be synthesized only if we add inducer or IPTG. This is something good about it. The regulation is being occurring inside the PET vectors, that as soon as the inducer is being added, the inducer is expressing the gene T7 RNA polymerase. This T7 RNA polymerase then binds to the T7 promoter which is present in the PET vector, leading to the synthesis of the target gene. And they have also introduced a Lac operator region which was blocking the synthesis of target gene, and IPTG comes and removes this blockade.
Ma'am, how leaky level synthesis is taken care of during overexpression using PET vector?
If the gene that we are introducing is toxic, then leaky level synthesis will be detrimental for the life cycle of the host cell. So what they have done, they have introduced another plasmid called P-lyse-S or P-lyse-E that synthesizes a T7 lysozyme which inactivates the basal level synthesis of T7 RNA polymerase that is getting transcribed from the P-Lac promoter. But the concentration of T7 lysozyme is so less that when there is overexpression of T7 RNA polymerase during IPTG induction, then it is unable to inactivate it. It is just doing the job to restrict the leaky level synthesis. So the problem while carrying on overexpression of a protein is that the protein sometimes behaves to be an insoluble. It comes in the insoluble part. So there are three different ways to convert an insoluble protein into a soluble part. First of all, what they do, that they carry on the induction at a lower temperature, 15 to 20 degrees centigrade instead of 37. And the second important pathway will be fusing the protein with some proteins like maltose binding protein. Here as you can see that MBP is able to overexpress the protein as well as it increases the solubility of the protein. And what another benefit that we get is that MBP is helping the purification of the protein, as if you pass the crude extract of the total protein and pass it through an amylose column, then it is only the MBP tagged target protein that binds to the column, and it can be eluted very nicely after addition of maltose. And the third important way wherein we can increase the solubility of a protein is by co-expressing certain chaperonin proteins like GRO-EL or DNA K, this chaperonin proteins, if co-expressed with the protein, they help in the folding of the protein and they increase the solubility of our desired protein. Now let's start the experimental part, that we are going to study the process of overexpression of a gene in E. coli using IPTG as inducer. The E. coli cell that we are using for overexpression of a protein is containing an expression vector that contains a gene called glutathione S transferase. It is under the control of a Lac promoter, which is the fusion of Lac and Trip promoter. It gets induced in presence of IPTG. We do have got Shine-Dalgarno sequences, SD sequence, which are the ribosome binding sites for the translation of the gene here. We also have got multiple cloning sites and terminator sequences. We need ampicillin for the growth of these cells because this plasmid contains a ampicillin resistance cassette. Next, we need IPTG for the induction of the cells to overproduce GST protein. Then we need cell lysis buffer, which is needed to carry on the lysis of the cells so that we can get the proteins. Next, acrylamide bisacrylamide solution, wherein it is present in the ratio of 29:1. Acrylamide is the polymerizing material and bisacrylamide is the cross-linking agent. So they get polymerized in presence of ammonium persulfate APS that initiates the polymerization event, and NN prime tetramethylene ethylene diamine or TEMED. These two components actually facilitate the polymerization of acrylamide and bisacrylamide to form the polyacrylamide gel. And in this gel, we need to introduce SDS. 10% SDS solution is needed, as the proteins that we are trying to separate out are separated on the basis of their molecular weight. They are denatured with the help of SDS and they have been imparted with enormous amount of negative charge. All of them are uniformly negatively charged. Next, we need here tris SDS buffer pH 8.8, which is the pH of the running gel. This SDS gel comprises of a stacking gel and a running gel. The pH of the stacking gel is 6.8 and its percentage is 5%, and the pH of the running gel is 8.8 and its percentage may vary from 10 to 30%. Next, we will be using molecular weight markers, protein markers. This will help us to get hold of the molecular weight of the unknown protein. We also have 5x sample loading buffer, which is needed for the equilibration or the lysis of the proteins. And we also have staining solution for the staining of the gel. We have got destaining solution to destain the excess stain so that we can visualize the bands of the proteins, and phosphate buffered saline is also a part of the gel loading buffer. Agarose is needed for the growth of the bacteria and LB broth is needed for the growth of the bacterial cells. Apart from this, the glasswares that are needed are conical flask, measuring cylinder and beaker. Distilled water is also requisite, and the most important instrumental requirements are protein electrophoresis apparatus. We will show you later on that how this apparatus looks like and how we are going to use it. We also need micropipettes and tips. 37 incubator as well as shaker are needed for the growth of the bacterial cells. We need centrifuge to centrifuge the cells. Crushed ice is needed for maintaining the stability of the proteins. Next, sterile loop is needed for streaking of the cultures, and microwave burner and hot plates are needed for carrying on the heating purposes. So, on the very first day, we will be provided with a stab culture of the E. coli cell bearing the expression vector. We will take a loopful from it and we will be streaking it on an LB amp plate and keep it inside an incubator for overnight incubation. Care should be taken that while streaking, you should do it perfectly to get single colonies on the next day. As we can see here that the single colonies are visible, we will take one single colony from such an LB plate and transfer it onto an LB broth, and we will allow it to grow for another overnight incubation in an incubator shaker at 37 degrees centigrade. Third day, from this saturated culture, we will take 1 ml of the saturated culture and inoculate it into a 50 ml LB broth, and this thing is kept inside a 37 degree incubator shaker for 2 to 3 hours depending on its OD reaching to 0.5, because it is supposed to be the stage wherein the bacteria or E. coli cell is residing in its mid-log phase. This is supposed to be the best stage for inducing overexpression of a protein. So before addition of IPTG as inducer, we will take out 5 ml of the culture into the tube and we will mark it as BI or before induction containing tube. And the rest of the culture is again kept inside the incubator and incubated, and we will add 0.3 molar IPTG onto it and kept it inside an incubator and incubate or shaken in an incubator for 2 to 3 hours. After that, the culture is taken out and 5 ml is collected in a tube. It is marked as AI, means after induction. Means we have got 2 tubes, one containing culture before induction, other containing culture after induction. Both of them are centrifuged. The pellet we are getting from these 2 tubes and supernatant is discarded. Next, the pellet is then suspended in 0.1 ml of lysis buffer. It is boiled for 20 minutes in a boiling water bath. Then it is spinned down and the supernatant is discarded. The pellet that we are getting is then suspended in sample loading buffer as we mentioned in the protocols earlier, and as a molecular weight marker is also treated with this sample loading buffer and phosphate buffer saline. This is the buffer tank with electrodes positive and negative. These are the glass plates wherein spacers are already there to incorporate space wherein gel can be casted. This is the casting frame wherein we are introducing the gelling material, that is acrylamide bisacrylamide SDS mix. After introducing the gelling material, we will put on the combs for making the wells. Then when this thing solidifies, we will remove those combs and this is called the casting strand. After that, when the gels have casted perfectly, they are put into the clamping frame and finally introduced into the buffer tank. All of them are loaded on SDS page. Loading is done like this and it is run on an SDS page and it is run at 120 volts. Initially, when we are running the gel, it should be done at 70 volts until and unless it reaches into the running gel. As I said earlier, that the SDS page comprises of 2 parts. One is called the stacking gel, other is called as the running gel. When the protein is loaded on the wells, it will first migrate through the stacking gel, then onto the running gel. Next, it is stained and destained. Then after destaining, we will be able to see the bands. So these are the steps that we are going to follow up, and it is connected to the power pack where from desired voltages are applied. It looks like this while running. Here blue dye is the bromophenol blue, which is called as the tracking dye. This helps me to understand that how long I am going to run this gel. Next, it contains a stacking gel as a running gel and the running gel. When the proteins are migrating to the stacking gel, its percentage is low. So we are allowing all the proteins to stack onto this part. After that, the proteins are entering into the running gel. Its percentage is almost 15% in this case, and herein we will see that proteins get separated on the basis of their molecular weight. Smaller proteins migrating faster, bigger proteins lagging behind. So we will run the gel until and unless the bromophenol blue dye or the tracking dye reaches the bottom of the gel. The result that we have got after running the gel, let's have a look what are the components of the lanes. Lane 1 contains the protein molecular weight marker, wherein these are the molecular weights of known proteins. Then lane 3 contains the BI sample, means the cell suspension before induction, and lane 5 contains AI sample, means after induction sample. So if I compare the lanes 3 and 5, I will see that most of the cases the proteins are the same, except for the protein in the region of 29 kDa. We will see the emergence of a GST or glutathione S-transferase protein, which gets synthesized only after induction with IPTG. This band is totally absent or absent in the before induction sample. So we can interpret the result as that the GST protein band 29 kDa is seen after IPTG induction, but it is barely visible in the before induction sample, and this indicates an increased level of GST protein production. So this is about the interpretation of a result, but you need to take certain precautionary actions while executing this entire experiment. Number 1, the cells which are to be grown, it should be grown following all the microbiological aseptic techniques. Secondly, while casting the SDS polyacrylamide gel, care should be taken. Gloves should be worn as acrylamide forms free radicals which are neurotoxins. And finally, when we are staining those gels or destaining the gels, it should be done in covered containers, as stainer and destainer both contain acetic acid, which produces tremendous amount of fumes. So to conclude, in today's lecture, we have understood the importance of overexpression of a protein in E. coli cells. We have understood that what features should be present in a host cell that is used for overexpression of a protein. We have also gone through the hurdles in the pathway of overexpression, and we have explained the entire pathway of protein overexpression using PET vector as the model expression vector and BL-DE3 as the model host cell. And finally, we have executed the experiment of overexpressing a glutathione S-transferase protein into utilizing a lactose promoter, which is being induced by IPTG.